Welcome to our first pre-lab lecture on cell staining. And in this lab that we're going to do this week, we are going to do some staining. And so I wanted to talk about some of the principles behind it. And so by the end of this module, you should be able to compare and contrast simple, structural, and differential stains, describe how stains interact with the cell envelope, and define the function of the gram stain and the acid-fast stain, and describe the function of the steps in a gram stain. So going a little bit deeper with the gram stain. And when we think about the way that staining works, it's important that we stain our cells most of the time because they are colorless. So the light is going to pass right through them. They may appear gray, possibly, if we don't stain them, or they may be photo bleached, which means that we can't see them at all without some sort of color. And most of our basic dyes are going to be positively charged. And that means that they're going to be attracted to the negative charge on the cell surface. And therefore, the cell will appear the color of the dye. So we have a couple of examples here of some basic dyes: methylene blue, crystal violet, safranin, and malachite green. Um, and safranin, for what it's worth, is kind of a pink or red color. Now, when you look here, I'm just kind of trying to demonstrate. We've got our cell, and then we've got our negative charges on the outside of that cell. Those negative charges are going to attract these positive charges that are representative of our dye, and they become tightly packed there. So just keep that in mind. Now, on the other hand, we have acidic dyes, as opposed to our basic dyes, and they're used in negative staining. Um, the dye in this case is going to be negatively charged. And so since the cell surface is negatively charged, then they're going to repel each other. And instead of just binding to each other, you're going to have this repulsion, and they're going to have a gap between the color and the cell. And so in this case, what you're actually doing is you're staining the background, right? So you're staining the negative space. Some examples here are nigrosin or India ink. Another term that we should know are mordants. They're chemicals that can be used basically to improve a dye's ability to stick. Iodine, alum, tannic acid have all been used as mordants. Now, when we think about these different staining techniques, um, they fall into a couple, like three main categories: simple, structural, and differential. And so our simple stains are just going to use a single dye, and then they're going to be used to determine some basic characteristics of the cell, like its size and shape. For example, when we look at these, we see these blue cells are going to be these longer ones are going to be some decently sized bacilli. It looks like there might be some smaller cocci over here. So the bacilli are the rods, and then the cocci are the circles. Now, our structural stains are used to try to identify specific structures. A common one is the flagellar stain. And what you do here is you're going to, actually, I've never done this, but my understanding is that it's a quite involved process where you have to repeatedly coat the cells, which includes the flagella, with the mordant, and then add your dye, a basic dye, which will stick to that mordant. And another structural stain is a capsular stain or a capsule stain, where you're going to have this capsule that surrounds your cell, and um, then you're going to um, stain the background, which is going to give you the ability to see that capsule. So in this case, you use a basic dye to stain the cell, and then you use a negative stain to stain the background. And then this space in between the two is where that capsule is. And that way that we're kind of finding that cell structure. Then there's the endospore stain. We use like malachite green for this, and you heat the malachite green into the cells over 10 or 15 minutes. And that malachite green is going to actually stain those cells nice and green. Then you add saffron into the cell itself, and you can see those. And so here we can see these different endospores that are forming inside these cells. It's a pretty nice cell stain or endospore stain. And so those are the three examples of structural stains that we might see. Now, there are two differential stains we're going to talk about. The first one is the Gram stain. The second one is the acid-fast stain. And this is a way to highlight differences in the cell walls of the bacteria. And so when we use a Gram stain, we use it to classify organisms as gram-positive or gram-negative, where our purple cells are going to be our gram-positive cells, and our pink cells are going to be our gram-negative cells. And the thing that I always try to point out uh, when we look at an image like this, if you didn't have this dark purple on here, you might call this like a lavender or a light purple, right? And so it's important to just really notice the difference between what is purple versus what is not, because that'll help you in the lab when you're interpreting results. Now, in this, what you do first is you heat fix your cells to a slide. And when you do that, they're going to be colorless, represented here by the gray. And that's whether gram-positive or gram-negative, cells don't have a distinctive color. They let the light pass through. They are just going to appear kind of gray. Then you're going to add a crystal violet stain, which is a basic dye. And both gram-positive and gram-negative cells will turn purple at this point. Next, we add iodine, which is a mordant. The mordant forms these crystal violet iodine complexes. So you basically bind up the crystal violet. They become these bigger complex molecules that are harder to get rid of. Next, we do a step of decolorizing. At this step, we're going to use acetone or ethanol. Or a mixture. And it's to wash out the stain from gram-negative cells. So if we decolorize properly, our gram-positive cells will still be purple, but our gram-negatives will be colorless. And then we hit with our counter stain, which is saffronin. And then, pardon me, at this point, the saffronin will turn the gram-negative cells pink, but the gram-positive cells will stay purple. And that's what that looks like together. And so to kind of get this, we got to think about our cell wall structure. And so in the cell wall lecture, you learn that the peptidoglycan of gram-positive cells is thick, and then the peptidoglycan of the gram-negative cell walls is thin, and they have that outer membrane, right? And so what happens when we add the crystal violet to the slide is it's going to actually bind in there with the cell walls of the gram-positive and the gram-negative. And that's why it'll look purple at this point. Then we add in our mordant, and that's going to form these complexes between the dye and the iodine. Still going to be purple at this point. You can't really see the color of the iodine. It's kind of like a yellowy brown, and the purple is just too strong to see anything like that under the microscope. So all you see is the purple. Now, when you decolorize, what's going to happen is it's going to dissolve the outer membrane of your gram-negative, and it's going to dissolve the dye out of your cell wall and cause some damage to the cell wall of your gram-negative. However, for the gram-positive, the cell wall is so thick that it might dissolve out some of the stain, but some is going to be left over. So if we looked at it at this point, the gram positive would look purple and the gram negative would look colorless. Now, lastly, we add our saffron in, which is our counter stain, and that's a basic dye, so it's going to bind to the negative charges of the cell wall. And in this case, this is the last step. Your gram negative cells are going to have um, the saffron in there. They're going to appear pink. Your gram positive cells are going to have saffron in as well, but they're still going to appear purple if you stained it correctly. And that's because the purple is just so dark that it washes out the pink. So there can be some experimental errors. Sometimes you can over decolorize a gram-positive cell, which causes you to damage the cell wall enough that you wash out all the crystal violet, and then they will stain like their gram negatives. Um, it's good idea to use like fairly new cultures, so 1 to 2 days old. And sometimes there are variations in the cell walls of organisms in general, right? And so some organisms just are easily decolorized, even though they're gram positives. Um, and then other times, like when you do have an old culture, that can lead them to be easily decolorized because you have a lot of dying cells. Now, the acid fast stain distinguishes between the gram positives that are acid fast and those that aren't. And because of that waxy mycolic acid layer, it actually retains the red color of the primary dye. But the non-acid fast cells don't have that waxy layer, so they do not retain the red. The way that this is done, just in very simple terms, is you use carbon fusion, which is kind of a red color, and you steam it into your cell, and you're going to add this acid alcohol mixture to rinse the sample, and then counter stain with methylene blue. And so you get this nice look where you're something like Mycobacterium tuberculosis, which is acid fast, is going to have this pink color from the carbonyl fusion, and then your other ones are going to have the methylene blue color. The acid fast staining is important for identifying the mycobacteria and the nocardia species. All right, so that is staining. You should be able to compare and contrast simple structural and differential stains, describe how stains interact with the cell envelope, and define the function of gram stains and acid fast stains, and describe the function of the steps in a gram stain. I hope you enjoyed. I'll see you in lab.